Hello everyone, and welcome to the lecture on plant molecular biology, where we will explore the fascinating field of plant tissue culture. In this session, we will cover the various culture methods used to propagate and study plants at the cellular level, focusing on the underlying principles and key techniques that drive this important area of research. The term plant tissue culture commonly refers to the in vitro and aseptic cultivation of any plant part on a nutrient medium in an artificial environment. In vitro work is carried out in a test tube, dish, or flask, outside of a living organism. Therefore, plant tissue culture techniques are also known as in vitro techniques. Different types of plant tissue culture include single-cell or cell suspension culture, embryo culture, callus culture, bud culture, organ culture, protoplast culture, meristem culture, and seed culture. Plant tissue culture is a valuable tool for studying morphogenesis, cell signaling, physiology, molecular biology, and biotechnology-assisted crop improvement. The impact of plant tissue culture technology on agricultural biotechnology is substantial. Two key concepts, totipotency and plasticity, are central to understanding the regeneration potential of plant cell culture. Totipotency refers to the ability of a single cell to develop into a complete organism, or regenerate into a whole plant, under suitable conditions, including its metabolic functions. Totipotent cells retain the full genetic potential of the mature organism from which they were derived, enabling them to divide and differentiate into a mature plant, when placed in an appropriate environment, or under favorable conditions. Plasticity refers to the ability of plants to alter their metabolism, growth, and development in response to changes in their environment. When plant cells and tissues are cultured in vitro, they typically exhibit a high degree of plasticity, allowing one type of tissue, or organ, to develop from another. Based on this principle, an entire plant can be regenerated. This regeneration of whole organisms relies on the concept that all plant cells, given the correct stimuli, can express the full genetic potential of the parent plant. Plant tissue cultures are generally initiated from sterile pieces of a whole plant. These pieces are called explants, and may consist of plant organs, such as leaves or roots, or specific cell types, such as pollen or endosperm. Several characteristics of the explant are known to influence the efficiency of culture initiation. In general, younger, more rapidly growing tissue, or tissue at an early stage of development, known as meristematic tissue, is most effective. When cultured in vitro, all the chemical and physical needs of the plant cells and tissues must be met by the culture vessel, the growth medium, and the external environment. The culture medium is one of the most important components of plant tissue culture, and a fundamental technique that makes plant tissue culture possible. The success of the culture medium largely depends on its chemical composition, as it must provide all the essential mineral ions required for plant growth and development. The composition of each tissue culture medium may vary depending on the nutritional requirements of different plant species. The supplementation of plant growth regulators in the culture medium is often necessary to trigger various types of growth and differentiation. Although explants may contain certain levels of endogenous growth hormones, or naturally synthesized growth regulators, it is often necessary to supplement them exogenously to elicit specific responses. Growth regulators are typically required in very small quantities, and both the type and amount needed in the medium vary depending on the plant species, tissue type, and stage of culture, such as callus initiation, somatic embryogenesis induction, shoot differentiation or multiplication, and shoot rooting. Physical factors such as light, temperature, and pH must also be maintained within acceptable limits. Some types of cultures require aeration and agitation to provide sufficient oxygen to the plant cells, and to enhance the availability of nutrients to all parts of the explants. Plant tissue culture is an essential component of plant biotechnology. In addition to the mass multiplication of elite plants, it provides a means to multiply and regenerate novel plants from genetically engineered cells. Plant tissue culture is associated with a wide range of applications, such as the rapid propagation of plants that are difficult to propagate, biosynthesis and production of pharmaceutical, medicinal, and other industrially important compounds, production of disease-free and resistant plants, germplasm conservation, and more. When cultured in an appropriate medium, typically containing both auxin and cytokinin, explants produce an unorganized, growing, and dividing mass of cells, called callus. Callus is usually composed of unspecialized parenchyma cells. During callus formation, some degree of dedifferentiation occurs, both in morphology and metabolism. Dedifferentiation is the phenomenon where mature cells revert to a meristematic state to produce callus. It is a transient process by which cells become less specialized and return to an earlier state within the same lineage. This indicates an increase in cell potency, meaning that after dedifferentiation, cells may possess the ability to redifferentiate into more cell types than they could before. One major consequence of this dedifferentiation is that most plant cultures lose the ability to photosynthesize. Redifferentiation is the ability of callus cells to differentiate into a plant organ, or an entire plant. Manipulating the ratio of auxin to cytokinin in the medium can lead to the development of shoots, roots, or somatic embryos, from which whole plants can subsequently be produced. Callus cultures can also be used to initiate cell suspensions, which are utilized in various ways in plant transformation studies. Callus cultures generally fall into two categories, compact or friable. They are produced when explants are cultured on an appropriate solid medium, with the correct concentrations of auxin and cytokinin. Auxin 2,4-D is commonly used to induce callus formation, as it can revert cells in the explant to a dedifferentiated state, and initiate cell division. The cells in compact callus are densely aggregated, and the callus may contain several small, prominent embryo-like structures. In contrast, the cells in friable callus are loosely associated, making the callus soft, and easily break apart. Friable callus provides the inoculum for forming cell suspension cultures. Callus proliferation can be maintained indefinitely in culture, if it is periodically subcultured in fresh medium, but the callus may lose its ability to redifferentiate after prolonged in vitro culture. Callus formation from an explant occurs in three stages. During the induction stage, metabolism is stimulated, and the cells in the explant begin to dedifferentiate and divide, while cell size remains unchanged. In the proliferation stage, cells divide actively, and cell size decreases. Cell division is primarily periclinal, and occurs towards the periphery, giving rise to wound cambial cells. During the final stage, cells differentiate through expansion and maturation. Vascular nodules, or meristemoids, are formed in the callus, from small groups of meristematic cells. These meristemoids resemble vascular bundles, and consist of xylem, phloem, and cambium. The nodules may give rise to root or shoot primordia, or embryos. When friable callus is placed in a liquid medium, usually with the same composition as the solid medium used for callus culture, and then agitated, single cells, or small clumps of cells, are released into the medium. Under the right conditions, these released cells continue to grow and divide, eventually producing a cell suspension culture. Agitation of the medium serves at least three functions. First, it applies mild pressure on cell aggregates, breaking them into smaller clumps and single cells. Second, agitation ensures the uniform distribution of cells, and cell clumps, in the medium. Lastly, the movement of the medium facilitates gaseous exchange between the culture medium, and the surrounding air. Cell suspensions can be maintained relatively easily, as batch cultures in conical flasks. They are continually cultured, by repeated subculturing into fresh medium, which dilutes the suspension, and initiates another batch growth cycle. After subculture, the cells divide, and the biomass of the culture increases, in a characteristic manner, until the nutrients in the medium are exhausted, or toxic byproducts accumulate to inhibitory levels. This is known as the stationary phase. If cells remain in the stationary phase for too long, they will die, and the culture will be lost. Therefore, cells should be transferred, as they enter the stationary phase. It is also important to determine the batch growth cycle parameters, such as the number of cells, nutrient or metabolite concentrations, and cell viability, for each cell suspension culture, to ensure the reproducibility of production. Potential uses of callus and suspension cultures include the synthesis of important biochemicals, or plant metabolites, by forming large cell biomass, rapid clonal multiplication and propagation of plant species, genetic engineering and production of transgenic plants, somatic hybridization, and the development of mutations for resistance to various environmental stresses, including pests and diseases. Organ culture is a type of tissue culture that involves isolating an organ for in vitro growth. Different types can be distinguished, such as meristem culture, shoot tip culture, root culture, anther culture, etc. Often, any plant organ can be used as an explant for the culture process. With organ culture, or when using various tissue components, the method is used to preserve the structure and function of the organ, allowing it to retain characteristics similar to those it would have in vivo. The organ continues to grow as long as it retains its physiological features. As such, organ culture provides valuable information on patterns of growth, differentiation, and development. Root culture can be established in vitro, using explants from the root tips of either primary or lateral roots, which can be cultured independently on relatively simple media. Since roots are indeterminate organs, their growth in vitro has the potential to be unlimited. Root culture can rapidly produce a large number of genetically uniform roots, making it ideal for studies on lateral root and bud formation, as well as the initiation of cambial activity and nodulation. Although the establishment of root cultures was one of the first achievements of modern plant tissue culture, they are primarily used for the production of useful substances, and have less impact on other areas of study. Hairy root culture is a type of plant tissue culture, used to study plant metabolic processes, or to produce valuable secondary metabolites and recombinant proteins, often in conjunction with plant genetic engineering. The tips of shoots, which contain the shoot apical meristem, can be cultured in vitro to produce clumps of shoots from either axillary or adventitious buds. This technique, also known as meristem culture, is extensively applied for clonal propagation in horticulture, agriculture, and forestry. Due to the small size of the propagules in culture, the in vitro propagation technique is also referred to as micropropagation. The process of micropropagation can be divided into three distinct developmental stages. Stage 1, the aseptic establishment of the explant. Stage 2, the multiplication of propagules, through repeated subculturing on a specific nutrient medium, and Stage 3, rooting, hardening of plantlets, and their transplantation into soil. Embryo culture is a type of plant tissue culture, used to grow embryos, isolated aseptically from the surrounding maternal tissues, of the ovule, seed, or capsule, under sterile and controlled physical conditions, in glass vials containing a nutrient solid or liquid medium. In embryo culture, the plant develops either directly from the embryo, or indirectly through the formation of callus, followed by the subsequent formation of shoots and roots. This technique has been developed to break seed dormancy, test seed viability, and produce rare species and haploid plants. It is an effective method, used to shorten the plant breeding cycle, by growing excised embryos, thus reducing the long dormancy period of seeds. Plant regeneration is a major outcome of plant tissue culture, based on the principle of totipotency. The ability to regenerate entire plants from selected, or genetically altered cells or tissues, is key to the broad biotechnological potential of plant cell cultures. In vitro plant regeneration is a process in which explants, after undergoing cell division and differentiation, form organs and tissues throughout their growth period. In general, there are two methods of in vitro plant regeneration used for practical propagation: organogenesis and somatic, or non-zygotic, embryogenesis. Organogenesis is the formation of organs, such as leaves, shoots, and roots, on a plant organ, typically of a different type, based on the inherent plasticity of plant tissues, and regulated by adjusting the components of the medium. Methods of plant regeneration via organogenesis rely on adventitious organs, arising either directly from explant tissues, or indirectly through an intermediate tissue, the callus. Both direct and indirect regeneration pathways give rise to meristemoids, which respond to factors in the system, to initiate a primordium, inducing new tissues, such as shoots or embryoids, depending on factors like the different ratios of hormones. In particular, the auxin to cytokinin ratio of the medium determines the developmental pathway the regenerating tissue will take. Typically, shoot formation is induced by increasing the cytokinin to auxin ratio of the culture medium, while the reverse is true for root initiation. An intermediate ratio of auxin to cytokinin promotes the proliferation of callus. Somatic embryogenesis can be defined as the process of embryo initiation and development from vegetative or non-gametic cells that differentiate into bipolar structures, mimicking zygotic embryos. Somatic embryos are formed from plant cells that are not typically involved in embryo development, such as ordinary plant tissues, and they are not surrounded by endosperm or a seed coat. The bipolar embryoids germinate to form both roots and shoots, in a manner similar to zygotic embryos, usually at a rapid rate, enabling the production of large numbers of propagules. The induction step involves reactivating cells to differentiate and develop embryos, which can be achieved through two methods: direct embryogenesis and indirect embryogenesis. Direct embryogenesis involves the development of embryos directly from the cells of explants, without any intermediary stages, such as callus formation. In contrast, indirect embryogenesis involves the formation of somatic embryos through several cycles of cell division, and includes an intermediate step of callus formation. Somatic embryogenesis is a multi-step regeneration process that begins with the induction of proembryogenic masses, and continues with the formation and maintenance of somatic embryos, maturation, and regeneration. Several signaling molecules known to influence or control the formation of somatic embryos include extracellular proteins, arabinogalactan proteins, and lipocytolipoglycerides. Temperature and lighting can also affect the maturation of the somatic embryos. In conclusion, there are several tissue culture methods, as different types of tissues require different conditions for the culture process to yield desired results. Plant tissue culture can be used for cloning, genetic modification of a specific plant, or simply to accelerate or increase the yield of the plant of interest. Although tissue culture is a complex process that requires a great deal of caution to avoid contamination, it is an investment that can offer rewards once you see the results. With that, this presentation has now concluded. It is hoped that the topic was engaging and informative. Thank you for your attendance and participation in this lecture.